Alright, everybody, welcome back to lecture. Technically, this is lecture 22, not 20, so let's get this out of here and change this to lecture 22. Yeah, I think this is based off of last year's kind of ordering. I'm pretty sure it was lecture 20 then when I took the class, but we are obviously not in last year, so yeah. Today's lecture is going to be on amino acid synthesis and all that jazz. Other than that, though, I mean, yeah, let's go ahead and get into it, shall we? So, pathways, all that jazz. We're going to talk about this stuff, generally speaking, and yeah. So you guys know this. Proteins are very important. Great. They make a lot of things, hormones, proteins, channels, all that jazz. But they play roles that are important besides protein synthesis. So things like tryptophan, phenylalanine, histidine, important in the immune response, important in neurotransmitters. So like serotonin and melatonin come from tryptophan. Phenylalanine makes all of the catecholamines like dopamine, epinephrine, norepinephrine. Histamine is important in like allergy response. Threonine is used to make collagen. Methionine and serine are very helpful in one carbon metabolism, making like nitrogenous bases. And then of course glutamine, glycine, aspartate are used in nitrogenous bases for making purines and pyrimidines. Also, if you've taken any neuroscience classes, you would know that glutamate is a neurotransmitter in the brain, and stuff like that. So these are all very important. So here we can talk about amino acid biosynthesis. So basically, all amino acids are derived from intermediates in all these pathways that we've talked about. And nitrogen, enters these biosynthetic pathways through amino acids like glutamate and glutamine. I think I switched it around, glutamine and glutamate. These are the most abundant amino acids in the body, period. And most bacteria and plants can synthesize all 20 amino acids, but mammals can actually only synthesize 11 of the 20. Here is like an image of what a general amino acid looks like. You guys should know this by now, so I'm not going to harp on that too much. So essential amino acids, these are amino acids that organisms cannot synthesize. So they need to be obtained from diet. And non-essential ones are ones that we can synthesize in adequate amounts. So we can synthesize in adequate amounts. And conditional ones, these double star ones, are amino acids that are normally synthesized but require additional levels from the diet. So like you can synthesize them but in small amounts. So essential ones are these. So these are all required to be achieved from outside sources, and these are all required. These are all things that you can make from the body. And then things like arginine, cysteine, glutamine, glycine, proline, tyrosine. These are all amino acids that you can that you would ideally get more help from your diet. You don't have to memorize all of these. There's too many of them. At least when I took the class, you did not have to memorize every single one of these, so I wouldn't be too worried about that. Could write on your note card if you're concerned about it, but I personally would not be too worried about memorizing every single one of them. What I would tell you to know and understand is how we get a decent number of these. So things like alanine, glutamate, and aspartate are very structurally similar to alpha keto acids that are in glycolysis and Krebs cycle. So these alpha keto acids can serve as the amino group acceptors to form amino acids. So for example, if we look at pyruvate, if I just change out this O here for an N, it becomes alanine. For alpha ketoglutarate, if I change out this O for an N, it becomes glutamate. For this guy here, if I change out this O here for an N, it becomes aspartate. And then finally, from the case of like glutamine and aspartate, if I was to change out this, if I changed out this O here on both of them to an N, right? That's not shown here, but you can see here, if I change out for an N, it becomes glutamine here, and it's not shown here for aspartate, but if I do the exact same process, it becomes aspartate, or asparagine, which is ASN, right? So you can see like how these things produce the amino acids that we're so used to seeing in our classes and in our bodies and stuff like that. So how do we transfer that? So how do we take this O and switch it out for an N? And the way that we do that is through these proteins called aminotransferases or transaminases. And so what they do is they basically just take the carbonyl group of a alpha keto acid and switch it out for an N. And yeah, so catalyzed by these guys, and they use PLP as a cofactor. So PLP, remember, is very common in decarboxylation reactions, but it's also extremely common in like carbon transfer reactions, as well as actually, sorry, not PLP. TPP is the decarboxylation one. I messed that up, sorry. TPP is the very common decarboxylation ones. PLP is important in like transfer reactions, like the transfer of the transfer of carbons, the transfer of amino groups in this case. So yeah, you can see here, if you take a alpha keto acid and an amino acid and you do an aminotransferase reaction using PLP, you get a new alpha keto acid and a different amino acid, basically. So here's an example of how this works. Here we have a glutamate and a pyruvate, and you're going to use a aminotransferase and using PLP. And what that does is it takes the ammonia group here and attaches it here instead. So it takes this and switches it right here. And so you're going to get alpha ketoglutarate and alanine. So this is an alanine aminotransferase because it creates alanine. It creates alanine. Likewise here, this is an aspartate aminotransferase because it makes aspartate. So you would take glutamate and oxaloacetate, take this N, switch it over here, use PLP aminotransferase, you make alpha ketoglutarate and aspartate. It's aspartate aminotransferase. This is certainly a slide. I don't think it's too important. All you really should know is that PLP is a versatile cofactor. It comes from vitamin B6. Hopefully you remember like vitamin B2, B3. It's like riboflavin and niacin, right, to make like FAD and NAD, stuff like that. B7, biotin, these are all things that are important for the MCAT if you're going to study for that. But just for this case of this class, you just know that vitamin B6 is going to be PLP. So if you have a deficiency in this, you're going to have some issues with amino acid production, as well as other things. But yeah, what you should basically know is PLP. I don't think this is all that important to know. I think knowing it's a shift-based linkage to a lysine residue is important. So like in the absence of a substrate, so when there's no amino acid bound, PLP is bound to the aminotransferase through a non-covalent interaction and shift-based linkage to a lysine residue. I think that's kind of important to recognize maybe, but I don't think it's super important. I think this is the most important part maybe. So upon binding substrate, the amino group of an amino acid covalently attaches to PLP. So basically this group here switches with the N group of an amino acid, which is why there's like a switcheroo here. So this carbonyl group gets switched out for this N on PLP. Here is the literal showcase of how that works. So it's like the ping pong mechanism is for sure something you should know. You should know that PLP uses this ping pong mechanism. So the amino group transferred to PLP forms a pyridoxyamine. So you can see here we have our PLP right here. And so when our amino acid comes in, normally it's bound to a lysine. See how this lysine is covalently attached here? But when the amino acid comes in, it displaces the lysine. And then you basically form this, like this imine right here, and then you hydrolyze it. And so that hydrolysis actually like breaks off this whole group. And so you end up with an OH and just this amine group. So that's the ping portion of this. The ping portion is this idea of you attach this on and then you kick it off. And the pong portion of it is the alpha keto acid reacts with this thing to form a new amino acid. So you form the imine again, and then you attack it with like a and yeah, so you form the imine again, you get like this water molecule leaving, so you get this plus imine here, and then once that is formed, you basically do a displacement reaction where this arginine acts as a base and attacks here and kicks off the group. So that's the ping pong mechanism. So hopefully that makes sense. I guess the ping mechanism is getting the N attached from one amino acid, and then the pong mechanism is grabbing a alpha keto acid and then attaching the N to that alpha keto acid. So remember, ping is for taking the N group of an amino acid, and the pong portion is taking an alpha keto acid and attaching that N that you just grabbed off of that amino acid. Here is glutamine synthesis. For this, you've got glutamine synthetase, which converts glutamate and free ammonia into glutamine. And it requires ATP because it's a synthetase. And this is found in the cytosol of the liver, skeletal muscle, and the brain. And it serves as the entry point for fixed nitrogen into anabolic reactions. Like if you need to take nitrogen and use it on something, you actually usually transport it in the form of glutamine. So this is also very active in things like catabolic stress. So this is the reaction. You take glutamate, you use ATP, you take ammonia, you use the enzyme, and you make glutamine, and you release ADP and PI. The reverse of this reaction is glutaminase, which turns glutamine into glutamate and free ammonia. And so in order for this reaction to occur, you actually have to transfer the glutamine from the cytosol to the mitochondria. And that's important because if you just put free ammonia in your body, it causes problems and a lot of things happen. So basically ammonia acts as a base and it actually deprotonates things and it actually causes all sorts of acid-base chemistry to get messed up. So from there, we utilize H2O as basically a hydrolysis reaction. So you utilize H2O and you release NH3. And so you can see here, this NH2 group is the one that ends up leaving. So water goes in, and it actually ends up attaching here. It's like a transesterification, essentially. When you learn this in OChem, I think you probably say that, taking an amide like that is not very stable, but just for the purpose of biology, you can just think of it like that. And, like I said this before, but asparagine synthetase is the same concept, where after you make glutamine, you actually use the glutamine to make asparagine. So that's where the N comes from. That's where this N comes from. It comes from glutamine. Okay, for synthesis of serine, and then cysteine, and eventually glycine, it's basically just a bunch of oxidations of 3-phosphoglycerate from glycolysis. So from glycolysis, remember when we do aldolase, we make G3P and dihydroxyacetone phosphate, and then we do 2,3-bisphosphoglycerate, right? And then we kinase that to make 3-phosphoglycerate, right? And then with 3-phosphoglycerate, what we do is we do a couple of things. So what we do is we do phosphoglycerate dehydrogenase, which oxidizes 3-phosphoglycerate, which basically turns it into a pyruvate, but instead of just having a carbon, a CH3 group on the end here, we actually have a phosphate group. So now we have 3-phosphohydroxypyruvate. And then from here, this is where the magic happens, if you will. We take a glutamate, and we use a PLP, so a phosphoserine aminotransferase, and we switch out this group here for a amine group. So now we have 3-phosphoserine, and you can see that this is literally just serine, except we have a phosphate group on here. So now we just use a phosphatase, so phosphoserine phosphatase, and then we have serine. And through some other reactions, all you have to know is that there's a replacement reaction where we take a sulfur and we'll switch it out for the O. That's how you get cysteine. And to get glycine, what you do is you actually use tetrahydrofolate, so with serine hydroxymethyltransferase. Also uses PLP. You use tetrahydrofolate, so you use this here. Tetrahydrofolate, you use PLP, you get this N5N10 methylene tetrahydrofolate, and you end up releasing water. So basically the OH group, so if I was to draw like this, just imagine this is serine over here, not cysteine. This group leaves as a water molecule, and then this carbon is attached to tetrahydrofolate. And so that's how you get glycine. So, one carbon transfer involves one of three cofactors. So there's three different cofactors you could use. And these cofactors are biotin, THF, and SAM. And so the main idea of this is that biotin always transfers things as a carboxyl group, right? So remember, malonyl-CoA, remember, that uses biotin because it's a carboxylation reaction. Pyruvate carboxylase, remember, uses biotin as a cofactor. Tetrahydrofolate transfers one carbon, like intermediates in like different oxidation states, so it doesn't really matter. But then SAM, S-adenosylmethionine, this is a derivative of methionine, always transfers one carbon groups as methyl groups in the most reduced state. So SAM is for most reduced, biotin is for most oxidized, and tetrahydrofolate is for everything in between. So biotin, I've talked about this before. Biotin is vitamin B7. It carries the most oxidized group of carbon, so carboxyl groups. And it does this through bicarbonate usually. So because you can't just exactly summon carbon dioxide from the air, if you will. So enzymes that use biotin, acetyl-CoA carboxylase, pyruvate carboxylase, propanol-CoA carboxylase. This you guys didn't really talk about too much, but this is for catabolism of odd-chain fatty acids. But remember, it's like that a bridge, right? It's like that the swinging arm that swings around. So here's acetyl-CoA carboxylase. How you see how like this binds here, and then it binds to this thing, and this thing swings over. That's basically how it works. Tetrahydrofolate is vitamin B9. I talked about this in terms of pregnancy, how it's important for like cell division and stuff like that. Mammals can't actually make this, and like I said, deficiency during pregnancy leads to neural tube defects. So essentially, it's a precursor for the formation of tetrahydrofolate. So folate is the normal version, but tetrahydrofolate is the super reduced form of it. And the formation of folate to THF is actually catalyzed by dihydrofolate reductase. And we talked about this before. It's very important for creation of a TMP, right? Because remember, for thymine to go from uracil to thymine, you actually have to add a methyl group. It's also important for epigenetic maintenance. So methylation, if you've ever heard of DNA methylation and stuff like that, those groups come from THF. And yeah, so sulfonamides, these belong to a class of synthetic antimicrobial drugs. And yeah, basically what these do is these inhibit synthesis of folate, and that actually kind of kills bacteria. So this is just kind of an example of a drug. Here is tetrahydrofolate. So basically what happens is here we have folate, normal folate. Using NADPH, we can reduce that down using dihydrofolate reductase into 7,8 dihydrofolate. And then we reduce it again, and that turns into tetrahydrofolate. That's basically the gist of it. And so here is kind of an image. I would honestly memorize these. I think these are helpful to memorize. I don't have them memorized anymore. It's been a long time. But essentially what's important about this kind of scheme here is, we talked about how THF carries one carbon units in different oxidation states. It's used to convert serine to glycine, and it's the main source of one carbon units, and stuff like that. So actually, sorry, I should rephrase that. So conversion of serine to glycine. So this serine to glycine thing, the one carbon unit, remember I talked about how that one carbon disappears from serine? It's not that it disappears to make glycine, it's actually that carbon unit is actually put into THF to form this N5-methyl THF. And this reaction is actually non-reversible, so you can't actually reverse this reaction once it's made. So here, let's see here, where do we start? Let's start with here. So we have tetrahydrofolate here, right? Starting from this, you have two options. You can either do serine hydroxymethyltransferase to turn serine into glycine. And what that does is it releases water, and it also attaches a carbon group. So it actually attaches a carbon group here. You can see this carbon group. So this is N5N10 because it's an N5N10 connection, tetrahydrofolate. And you have two options from here. You can either reduce it again through N5N10 tetrahydrofolate reductase using NADPH. That makes this CH3 group, right? You can see how that's really reduced, right? This is kind of oxidized because we've got these N groups, right? We've got two N groups, and N's are like similar to O's in how they would be like an oxidizing group, if you will. They pull electron density away from the C, so the C has like a partial positive. And so you can see how this group is more oxidized than this group, right? So this is a CH3, very reduced. This has the N connected to it. It's one thing oxidized. Or you can oxidize it again. I feel like this is... You can oxidize it again using NADP+, and you can go this way to make this. And so you can make this, and basically you're oxidizing this, and so you make this aldehyde, if you will. And what is important to recognize about this, actually, is that, like I said before, this is irreversible. So once you go here, if you go straight from tetrahydrofolate and you take formate and you use this with the N10 formal tetrahydrofolate synthetase, if you straight make this, you can't go back, right? You can't go back this way. Is the main thing I wanna properly inform. You can go. I think you can go back this way and then back this way. But you can't just go this way and then back this way. That's not possible. So this is a slide that I would try to remember. I would try to remember all these enzymes and how they work. Hopefully it's relatively intuitive. Basically with tetrahydrofolate, you use the serine hydroxymethyltransferase. You get this N5N10. And from there, it's just this same enzyme, basically. So you have an N5N10 reductase or a dehydrogenase. And you either do a reduction reaction or an oxidation reaction, where you basically do that, right? So now we talk about SAM, which is S-adenosylmethionine, and it is the preferred cofactor for biological methyl group transfers. So, we talked about THF, but the one that actually does the transferring of these methyl groups is actually SAM. So this is very important for the synthesis of epinephrine and lipids. It's very important for methylation, and also is very important for like DNA methylation specifically. So histone methylation and DNA methylation, I believe. It's also basically the way that this is synthesized is from ATP and methionine. This should not be a shock to anybody, though. So the methyl group is subject to attack by nucleophiles. So this plus charge here allows us to actually attack the methyl group and kick off these electrons here. So that's why it's kind of a good donator of methyl groups. So yeah, the destabilizing sulfonium ion, this plus charge here, increases the reactivity, which makes it way more reactive than the N5-methyl tetrahydrofolate, so way more reactive than this guy here, right? This guy is not super reactive, pretty stable, I would say, so he's not super reactive, but making this sulfonium ion, this ion here, makes it very reactive. So here's how you actually form SAM. This is very important to remember, I would say. Starting from methionine, what you basically do is you use the electrons from methionine to attack the carbon 6 of this ribose here, and that kicks off all the ATP. It kicks off the 3 phosphate groups, and those are hydrolyzed. So you hydrolyze it into a PPI and PI. Those are both hydrolyzed. So this pyrophosphate group is then hydrolyzed later, right, to release some energy. And so then you end up with this thing. This is done through methionine adenosyltransferase. And then when you end up with this thing, you now have this sulfonium ion. And so now you can just use a variety of things, like there are a bunch of methyl transferases, and, they are able to take the CH3 off of it. And then from there you have this S-adenosyl homocysteine, which is, adenosine basically attached to methionine, but it's not really methionine because it's technically cysteine, but there's actually 1 carbon extra, so that's why it's called homocysteine. But then you use a hydrolase on it to remove the adenosine, so now you can recycle this adenosine, and now you have homocysteine. And then from homocysteine, we actually use this cofactor called cobalamin, so coenzyme B12, and you actually have methionine synthase, which what this does now is using B12, it's able to use the most reduced form of this, so N5-methyl tetrahydrofolate. It's able to remove the CH3 group and put it back on methionine, and it makes THF as a byproduct as well. All this to kind of say, you know, these are things that you should know. I would try and remember these. Methionine synthase, very important to remember that from homocysteine, we use B12 and we use N5 tetrahydrofolate. So basically the cofactors is B12, but the substrate is the methyl tetrahydrofolate. That's how you make methionine, right? And then, of course, remember these enzymes as well. So now we talk about, you know, conversion of serine to glycine. We've talked about this a little bit. I'm not going to harp on this too much. I would just say that, you know, serine hydroxymethyltransferase requires 2 cofactors. So it requires PLP and THF. And, you know, PLP-dependent cleavage of the alpha-beta carbon-carbon bonds. And so, essentially what's happening here is, for this, all we're doing, right, is we are removing this carbon here and the water molecule. And so I'm trying to see if they show it. But it doesn't look like they really show it. Essentially, like this arrow is showing, it's the cleavage of this bond right here. That bond is cleaved. And so THF is a soluble factor. It accepts the methylene carbon. So it accepts this whole carbon, basically. It accepts this carbon, and the water is released as a result. So once you do that, you form this. So PLP-dependent cleavage of this bond, like I said. The THF accepts this carbon. So remember that folate is just this without this CH2 group, but THF then accepts this this group, and then from there this whole group is released, and then you basically cleave off this H2O, this OH, put an H on it and make it water, and then you release glycine as a result. And so then after you make this, you can go in either direction. Remember, you can either oxidize it this way or you can reduce it. So you can either oxidize it or you can reduce it. And then you can use this here to make methionine, and then you can use that to methylate stuff. Remember, so okay. So here's some of the linkages. That, you know, this is like a helpful thing to know. I think, you know, how do all these things link together? And so remember that, you know, N5, N10, right? This is helpful in purine synthesis. Also used in amino acid synthesis, so, you know, you do that, and, you know, from here, once you've, you know, maybe used it for one of these 2 things, it returns to N5-methyl tetrahydrofolate, and so methylene, remember, is a CH2 group, methyl is a CH3 group, so maybe you use this to, you know, to make a purine or whatever, and then you return as N5 THF, methyl THF. This could be used by methionine synthase with B12 to make methionine, right? Which can then be used to make SAM, and then this SAM can be used to methylate things. And then, you know, from there you would get homocysteine. You know, you can see this whole, like, series of events that we're doing, right? So it's just a lot, right? So it's a lot of stuff going on here. So remember, from THF, we use serine to grab that methyl group, the methylene group. We make N5N10. From N5N10, we can do DNA stuff. We can do amino acid stuff. And then from the N5N10, we can then reduce it or we can oxidize it, right? If we reduce it, we make N5-methyl, and then we can use N5-methyl with methionine synthase to make methionine, right? Using vitamin B12 as a cofactor. With methionine, we can make SAM, and then with SAM, we can make S-adenosyl homocysteine, right? And then, we can use... and then once we cleave it, we make homocysteine, and that whole process restarts, right? And technically, there should be a spot in here where we talk about maybe you use a methyl group, right? So, like, SAM's used to methylate things, right? So maybe you methylate it. That's how you get the S-adenosyl homocysteine, basically. Right? Where is it? Right, see here. It's like the S-adenosine homocysteine, right? There should be a methylation step in the middle here. Like, so you see this whole step here? That is not shown here. That should be right here. Right, there should be a step of methylation where like you can methylate something here. So, yeah. Significance of cobalamin. It's required for a couple of reactions, rearrangement of these two things, and also conversion of homocysteine to methionine. This is the important one. If you don't have B12, what you end up getting is, you don't have, this reaction doesn't occur. And so what you end up getting is, remember that formation of N5 is irreversible, right? Remember, so basically you build up a lot of this stuff, which depletes all of the methylene version, and so you would get a whole bunch of problems. So, where is it? If you don't have B12, right, this does not work. So you don't actually make any of this. So you end up with a bunch of this, and you have none of this because you need this to go to this in order to do that. I think is how you should think about that. So buildup of this thing, and yeah, it leads to like a lot of problems because you need like THF and all that stuff, and you need N5, N10 as well to do other things in the body. So yeah, other than that, that's actually the end of this video. So I hope it was helpful. I know I kind of rushed the last couple of slides. Gosh, my voice. But if you have questions, feel free to ask. I'll be at the IC. Planning on having an exam review workshop on Tuesday, so stop by for that if you're interested. But other than that, have a good one and happy studying.